Hello, everybody, and welcome to the HPTN 083 Study Update Press Briefing. I am Eric Miller, Senior Communications Officer for the HIV Prevention Trials Network. At this time, all participants should be in listen mode only. Also, please stop your video. This will help with the call quality. Later, you will have the opportunity to ask questions during the question and answer period. It's my pleasure now to turn today's program over to Dr. Myron Cohen, HPTN Co-Principal Investigator and Director of the Institute for Global Health at the University of North Carolina in Chapel Hill. Dr. Cohen?
Good morning to everyone. Thank you so much for joining us this morning. There's a lot of people on this call, which is inspiring to me because it indicates interest in what we think is a really important observation or discovery, if you will. I think that my job is really to turn this over to Dr. Landovitz to summarize what has already been expressed in press releases, to say a few words. He and Dr. Beatrice Grinstein are the principal investigators that led the HPTN 083 study. And I suspect you'll have several questions for him. So Dr. Landovitz, why don't I turn it over to you?
Actually, Dr. Cohen, that's a correction. At this time, we would like to hear from Dr. Carl Diefenbach.
Yeah.
Director of the Division of AIDS.
Okay.
Dr. Diefenbach?
So thank you. Good morning, everybody. And as the representative for the National Institutes of Allergy and Infectious Diseases in the NIH, I just want to acknowledge the incredible partnerships that were necessary in order to complete this study. Working with ViiV Healthcare and Gilead Sciences, we were able to perform this study, as Dr. Cohen has said, by the HIV Prevention Trials Network, so ably led by Beatrice and Rafi. So we'll hear about that in a moment. But again, the study would not have been possible without the co-funding from ViiV as well. So I want to just acknowledge our partners as we move forward. And now I'll turn it back to Eric.
Thank you, Dr. Diefenbach. So now we will hear from Dr. Kimberly Smith, Senior Vice President, Head of Research and Development for ViiV Healthcare. Dr. Smith?
Thank you, Eric. This is Kimberly Smith, and as you heard, I am the Head of Research and Development for ViiV Healthcare. I am, just on behalf of the company, I want to thank Carl and Mike for the partnership with HPTN and AIDS, and certainly thank Rafi and Beatrice, the PIs of the study, really the whole study team. This has been a tremendous collaboration, and we could not be more excited with how effective the study went, how well it was run, and ultimately we are thrilled with the results. Rafi will go over the results in some detail, but I think what I would like to just emphasize is that we're thrilled not only because cabotegravir was shown to be highly effective at preventing HIV acquisition in men who have sex with men and transgendered women who have sex with men, but that we did this study in a population of individuals who often are not included in these trials. This represents in the U.S. more than 50% of the U.S. enrollees are men, Black men who have sex with men, who are the most impacted population in the United States. And around the world, young men who have sex with men are disproportionately impacted. And this study is strongly representing that population, with the majority of individuals being in that younger age group. And also the inclusion of 12% transgendered women is also a critical population, really disproportionately impacted. So we're so proud to see CAB be able to demonstrate efficacy in this population. And we look forward to the continued collaboration with HPTN on the 084 study. Hopefully we can generate strong efficacy data in women as well. We really look forward to that. And again, we look forward to moving cabotegravir forward and filing for approval, not only in the United States, but actually around the world, to add this to the toolbox of options that we have available for HIV prevention. And I'll stop there.
Thank you, Dr. Smith. Now we will hear from Dr. Raphael Landowitz, HPTN OA3 Protocol Chair and a Professor of Medicine in the Division of Infectious Diseases at the David Geffen School of Medicine at the University of California, Los Angeles, and Associate Director of the UCLA Center for Clinical AIDS Research and Education. Dr. Landowitz?
Thank you and good morning. And again, I just want to echo my thanks to you all for being here this morning to discuss these exciting results. I'm going to first begin by briefly reviewing the study design of HPTN 083. HPTN 083 is a phase 3 randomized multi-center double-blind clinical trial that was designed to evaluate the efficacy and safety of long-acting injectable cabotegravir, also referred to as CAB-LA, to prevent sexual acquisition of HIV in cisgender men who have sex with men and transgender women who have sex with men who are at increased risk of HIV infection. The study compares the effectiveness of CAB-LA to daily oral Truvada. As you all are aware, Truvada has been shown to be highly effective for HIV prevention when taken as prescribed in a variety of populations and clinical contexts. In HPTN 083, study participants were randomized in a 1-to-1 ratio to one of 2 study arms. One arm provided active cabotegravir and a Truvada placebo. The other arm provided active Truvada and placebos for cabotegravir. The cabotegravir or its placebos were administered daily by mouth for 5 weeks and then via intramuscular injection at 8-week intervals after an initial 4-week interval load. Participants in both arms were then offered open label daily oral Truvada for 48 weeks after their final injections. I'm now going to describe the study population that was enrolled into HPTN 083. A total of 4,570 HIV uninfected participants were enrolled into the study between December 19th, 2016 and March 16th, 2020 at 43 sites in Africa, Asia, and North and South America. These enrollments included 37% of its enrollees from the United States, 43% from Latin America, 16.5% from Asia, and 3.5% from Africa. A total of 4,565 participants were included in an analysis that was reviewed by the NIAID-NIH Data Safety Monitoring Board on Thursday, May 14th, 2020. The protocol team worked very hard to achieve representation, as Dr. Smith just noted, from some very important groups for HIV prevention. The mean age of our study participants was 28 years. The majority, 66% of our participants, were under the age of 30. Fully 40% of our participants were under the age of 25. 12%, as Dr. Smith again mentioned, of our enrollment were transgender women. 50% of our enrollment from the United States, again as noted by Dr. Smith, self-identified as African American or Black. As I mentioned, a pre-specified interim review by the NIH multinational DSMB occurred on Thursday, May 14th, 2020. At that review, the DSMB concluded that the blinded study had reached its objective and they recommended stopping of the blinded portion of the study. The DSMB considered in its analysis the preliminary finding of 50 incident cases of HIV infection. These 50 infections are included in the intent to treat analysis and are all infections that occurred after enrollment and are included regardless of whether participants continued taking their randomly assigned study products. The overall HIV incidence among all study participants in HPTN 083 was 0.79%, suggesting that both cabotegravir and Truvada are both highly effective for HIV prevention in the HPTN 083 population. 38 incident infections were detected in participants assigned to the Truvada arm, for an incidence rate of 1.21%. 12 incident infections were detected in participants assigned to the cabotegravir arm for an incidence rate of 0.38%. That is, we observed roughly 3 times the number of incident HIV infections in the Truvada arm than we did in the cabotegravir arm. The hazard ratio in the cab versus Truvada arms was 0.31 with a nominal 95% confidence interval of 0.16 to 0.59. The interpretation of this finding is that cabotegravir was highly effective for HIV prevention. These results clearly ruled out the pre-specified non-inferiority margin of 1.23 with evidence approaching superiority pending our final analysis. Based on these efficacy findings, the independent DSMB recommended that the blinded study component be stopped early, the study participants unblinded, and results released to the scientific and broader stakeholder communities. I am now going to describe some additional results from the study, including some safety results. In a random sample of approximately 400 HPTN 083 participants receiving Truvada, plasma tenofovir concentrations and dried blood spot intraerythrocytic tenofovir diphosphate concentrations were measured. 87% of the plasma samples analyzed contained detectable concentrations of tenofovir. 95% of dried blood spot specimens assayed contained detectable concentrations of tenofovir diphosphate. This suggests that the HPTN 083 study population was even more adherent to daily oral Truvada than had been planned for in the original study design, likely conferring an even higher level of protection against HIV infection for Truvada recipients than had been planned for in the original study design. Concentrations of tenofovir in plasma and tenofovir diphosphate in red blood cells for all those who acquired HIV infection after being assigned to Truvada are currently being evaluated. Concentrations of cabotegravir in plasma for a subset of all participants and for all those who acquired HIV infection after being assigned to cabotegravir are also currently under evaluation. Resistance testing for the incidence HIV infections is also in progress. An analysis of the timing of the incident infections in participants receiving cabotegravir relative to their last known clinic-provided injection is also underway. From a safety perspective, both cabotegravir and Truvada were safe and well tolerated in HPTN 083. Most adverse events were mild or moderate and well balanced between arms. Injection site reactions, pyrexia, and hypertension were more commonly observed among participants in the CAB arm than in the Truvada arm. Injection site reactions were reported in 80% of participants in the CAB arm and 31% of participants in the Truvada arm who were receiving a placebo cabotegravir injection. Most of these injection site reactions were mild or moderate pain and or tenderness. Among participants assigned to cabotegravir, 46 participants, or 2.2% of those who received at least one injection, discontinued their injectable study product due to an injection-related adverse event. No participants assigned to the Truvada arm with the placebo injections discontinued injections for this reason. The severity of injection-related adverse events was significantly associated with increased odds of permanent injectable study product discontinuation, but again, to emphasize, these discontinuations were extremely rare. I am now going to review the immediate next steps for the HPTN protocol, HPTN 083 protocol. All of the institutional review boards and ethics committees overseeing the study, site investigators, and study participants are being notified of these results as soon as possible. Participant randomized treatment assignments will be available to site investigators this week. The study team is preparing a letter of amendment that will allow participants to stop taking their placebo medications, but continue on their currently assigned active PrEP product in the protocol. CAB-LA will be offered to all study participants as soon as that medication can be made available at sites. All participants currently on the study will be offered and encouraged to continue to be followed on the study. Participants choosing not to remain in the study will be referred for the best locally available HIV prevention services. A full revision to the protocol and protocol amendment will follow this more immediate letter of amendment to this ongoing study to allow for a more prolonged follow-up on open label CAB-LA. Additional details of that full amendment and open label extension will be forthcoming shortly. The United States Food and Drug Administration has been made aware of these results, and conversations with other regulatory agencies are being planned. We look forward to presenting these results in peer-reviewed settings at upcoming conferences and in manuscript form as we continue to finalize the primary analysis and begin work on multiple important secondary analyses. I would like to reiterate something that Dr. Smith already mentioned and Dr. Diefenbach referred to. Very importantly, HPTN 084, a parallel trial in cisgender women in sub-Saharan Africa, asking a similar question of the efficacy for HIV prevention of long-acting cabotegravir compared to TDF, FTC, or Truvada, opened to enrollment approximately 1 year after HPTN 083 and has nearly completed enrollment with approximately 25% of its planned person years accrued to date. The study has been reviewed by the same NIH multinational DSMB, most recently last Thursday, May 14th, 2020, and will be reviewed again when more data are available at the latest by November 2020. At the current time, that DSMB, who had earlier the same day reviewed the HPTN 083 data, recommended that at this time the HPTN 084 study continue without change. Additionally, adolescent bridging studies are poised to begin enrollment to provide critical safety data in adolescent and young adult populations in support of regulatory submissions for youth. Qualitative sub-studies of both 083 and 084 are continuing to explore barriers to sustained and consistent use long-acting cabotegravir. That concludes the results portion of this presentation. I would like to echo Dr. Smith, Dr. Diefenbach, and Dr. Cohen's thanks for the incredible partnerships we have had from FHI 360, the SDMC, between the HPTN network, Gilead and ViiV, and the NIH, DAIDS, NIMH, NIDA collaborations, as well as all of our sites and our participants that have led us to this exciting result. Thank you very much.
Thank you, Dr. Landovitz. At this time, we will unmute the lines for a question and answers period. When asking a question, please remember to share your name and news organization. So with that, let's go ahead with the first question. James?
Hello.
Please go ahead.
Hello, this is James Queeley from Morgan Stanley. A couple of questions. So can you put this into context for us? So we've now had this trial for 2 months, cabotegravir versus Truvada. We've also had, I think it was the Discover trial, which was Truvada and Descovy. So how, when you're looking at HIV patients who, or HIV at-risk people who could be candidates for PrEP, how should we now think about these three therapies, and who would you select to take these therapies based on the data you've seen so far? Also, in terms of the benefits shown, there's a 0.31 hazard ratio, 95% confidence intervals, both below 1. What are you missing really for statistical significance? Is it just the case that the final calculations and statistical tests haven't been done yet, or is it a case that there's something else missing? Thank you.
Yeah, thank you for those questions. Let me sort of answer them sequentially, if I may. The first, I believe, was a question of how one thinks about the 3 products, only one of which has global regulatory approvals, the other approval in the United States, that's Descovy, and CAB, of course, does not yet, has not yet gone under review for regulatory approval for pre-exposure prophylaxis. So I think it's premature to speculate, but I will say that it's clear that more choices of modality should the injectable product receive regulatory approvals, because it's clear from previous work that daily oral tablets or even event-based tablet dosing is challenging for some individuals. And we look forward to providing hopefully providing additional mechanisms of delivering highly effective HIV prevention to at-risk individuals. I think we're all very excited for the future results of the HPTN 084 study, which will hopefully recapitulate and confirm these results in cisgender women, a much needed data gap for additional options for prevention. Let me then move on to your question about the hazard ratio of 0.31 and the confidence interval appearing to exclude 1 and what's missing for superiority. I would just remind everyone, and this is a statistical nuance, when I quoted the confidence interval around that 0.31, I used the term a nominal 95% confidence interval, and that's because this was a very early look at the data at which the study was stopped. You may recall the original study design was predicated on 172 HIV infections being observed, and 3 pre-specified interim analyses were in the original protocol design. The first, when 25% of those endpoints had occurred, and this was therefore the first interim analysis. So a much more rigorous p-value and confidence interval was required both to declare non-inferiority and would be required to declare superiority at this extremely early time point. We are still doing final analyses of this interim data set, but at this time, we have an incredibly strong non-inferiority statistical result that approached superiority, but did not cross that superiority bound. The final analysis is still pending, and we hope to report that soon.
Excellent, thank you very much.
Thank you, James, for that question. Also, would like journalists know that the Zoom group chat feature is available to you if you prefer to ask your questions via the Zoom function. I would just ask to please include or share your name and news organization if you use that Zoom group chat feature. I don't see any other hands raised, so are there any other journalists who would like to ask a question of our speakers?
Yes, I would. Steen Stovall of Script. Hello?
Go ahead, please.
Hi. Yeah, congratulations, first of all. Obviously, now it's the end users who are keen on knowing when this might be available. So a question with that perfect world scenario, presuming you're now going to ask whether the phase 2 trials data is sufficient to avoid going to a phase 3. So Kimberly, just in a perfect world scenario, when do you hope to have this product available on the market?
Thanks, Sten. So it is a phase 3 trial. So obviously, this would be considered a pivotal trial. We will be in discussions with the FDA about a data package for a file. In an ideal world, we would be able to file for men and women together, because we do think that that is very, very important. It will depend on the timing of the delivery of the 084 study data, whether or not that will be possible. But I can tell you that we are working very closely with our partners at the Division of AIDS to try to pull together the data package and put together a file as soon as possible. I would not be, it would not be right for me to try to put a time frame on that yet, because there's yet still a tremendous amount of work to do, as Rafi, Dr. Landowitz laid out quite clearly. This is hot off the presses, and so there's a lot of data, a lot more analysis to be done, and a lot of data to be pulled together to answer every question that will come up. But we, again, it is a high priority, and we want to get it filed not only in the US, but also in Europe and around the world as soon as possible. Thanks,
Kimberly. Other question would be, okay, looking at it as a monotherapy preparation, what are your hopes for that going forward?
So this is a monotherapy. So it was only cabotegravir compared to Truvada. So there was no two drugs with the long-acting arm. It was only cabotegravir. Thank you. And last question, what about the CRL that came out, complete response letter? You have any worries about that, or we're just going to assume that'll be sorted out with time? So as we mentioned previously on our results call, our intention is to resubmit the data on CMC. As you recall, the reason for the CRL was related to chemistry manufacturing controls. And we are working through those issues with the FDA, and that is our expectation that we will resubmit by mid-year, and that would mean an approval late 2020 or early 2021. Thanks, Kimberly.
Thank you, Steve, for those questions. Anyone else like to ask a question? One of our speakers.
Hi, Lisa Urquhart from Environment Advantage. I've got a few questions. The first question is around the trial length. Is it true that the trial was halted due to the impact of COVID and people actually attending meetings, and that was one of the impacts in terms of proving superiority? My second question is, do you have any theories around the 12 people that were infected? One of the things I've read is the fact that it might be around body mass index, with some of the lighter subjects expelling the drug faster from their bodies, and also the fact it might have been around the 5-week oral run-in time. And then my final question is, as a monotherapy, are there any concerns around resistance?
Thank you for those questions. Let me try to address them in order. Was the trial stopped because of COVID? No. Obviously the COVID pandemic is affecting global populations, and to that extent, trial participants and sites are not different. We were concerned that COVID had the potential to disrupt study-related activities, and so we brought that to the attention of the DSMB before the most recent review to make them aware that this data cut that they were seeing was likely to be the last data cut that we could be assured was prior to any disruption that would be attributable to COVID. And then we did observe that some of our trial sites were needing to be closed because of the COVID pandemic subsequent to those data cuts. So the decision by the DSMB to stop the trial at this time was still a purely statistically based observation that we had crossed a non-inferiority boundary. The original study design would have instructed, the original study monitoring plan would have instructed the DSMB to only stop the study early for superiority at this particular time, but to continue for non-inferiority. this pre-DSMB review conversation that we all had with the DSMB did in fact instruct them, because of the COVID epidemic and its anticipated effects upcoming, that we all would be satisfied and comfortable stopping the study at this time for non-inferiority. And so the DSMB made the decision with that guidance and the independent observation that non-inferiority had been resoundingly met and superiority approached, that now was the correct and appropriate time to recommend stopping of the blinded portion of the trial. With regard to the 12 infections that were observed in the cabotegravir arm, this is, of course, a critically important question, and we're actively looking into what the characteristics are of these participants, as well as the timing of these infections relative to their last known administration of cabotegravir, as well as their plasma cabotegravir concentrations. I think at this point it is premature to speculate what contributed to those infections until we have more information. The question about monotherapy as a PrEP agent and resistance is, of course, a critical question. And again, we are working quickly and diligently with our laboratory partners and our sites to get the appropriate samples to analyze the resistance patterns and contexts of the infections that were observed in both arms. And we look forward to presenting those data. I think that's all we can say at this point.
If I could just add one point to Rafi's excellent summary. I think the DSMB made the decision to stop the study, as Rafi said, with that, given that criteria that they could stop on the basis of non-inferiority. However, the decision to recommend that individuals who are on the Truvada arm be offered cabotegravir is on the strength of the cabotegravir data.
Okay, thank you very much.
Thank you all there. Next question. I am not seeing any hands raised or any questions in the group chat. Would anyone else like to ask a question of the presenters?
If you're speaking, it's mute. You may be on mute.
By the silence, I would take it that there are no questions at this time. I would remind journalists that are on this call, in the event that they do have questions after this call ends, to please refer to the media advisory and press release that were shared earlier this morning, and to contact Kevin Bokach, communications officer, and he will be glad to put you in touch with the speakers who have presented today, as well as those mentioned in the press release. So I will just go around one more time and ask, are there any other questions before we end this call?
I do have some. Can you hear me?
Yes, please.
Yes, this is Gus Cairns from AIDS Map. Sorry, I have several, but I've already asked Myron Cohen to answer some of them. A couple, just two. Firstly, there was going to be a kind of tail-off phase in the study to cover the long tail of cabotegravir, with people offered oral TDF for 48 weeks at the end. That's now not happening, or will it still be offered to people as part of the study who wish to continue taking the pills?
Thanks for that question. So I think it is actually still happening, because we have some participants who have completed the injectable phase already, and they've already begun that oral tail. We also had some participants who stopped their injections early and transitioned to that phase early. So that's been in process and implemented. The context of whether or not it will still be required in its current form, or now that it has been shown that cabotegravir has protective efficacy, oral cabotegravir could potentially be used to cover that tail if someone stopped injections, are all under discussion. So it would be premature for me to definitively state the way forward, but I think the exciting conclusion is that cabotegravir clearly has high levels of HIV prevention efficacy, so it's likely we will have additional options, but we need to interrogate further if coverage of that tail is even something one needs to do. I think to be conservative, lots of discussions need to take place and lots of analyses before that can be definitively concluded.
Okay, thank you very much. If anyone's got any other questions, that's fine, but I also have one more I'd like to ask if possible.
Please, Gus, go ahead.
Yeah, okay, thank you. The adherence of the people taking the TDF-FTC, that was 87% had more than 0.31 nanograms a milliliter of drug in their blood. But as far as I can see, that's only the level you'd associate with 1 pill a week. So their adherence was not particularly high. Do you have a figure for the average adherence or any other figure other than that one?
I don't. We do have dried blood spot information. Actually, I take it back. I think we do have an average. Hold on 1 second, if you don't mind. Kim, if you have it in front of you.
So 75% had levels consistent with daily therapy.
Oh, that's very interesting. Okay, thank you. I'll put that in. Glad I asked. Okay.
Thank you. Gus, was there any other follow-up briefing on that question?
We do have dried blood spot data on that same adherence sample of 400 Truvada treated participants. But again, I only have top line detectable levels. I don't have the average, more traditional categorical analyses that we're more used to seeing for dried blood spot analyses. We're working on that.
Thank you, Dr. Rafi. Any other questions from the journalists before we wrap things up here for the morning? Okay, then. Well, it looks like there are no questions. As I said previously, a journalist, if you do have a question or questions after this call ends, please refer to the press release and to the advisory you were sent this morning for contact information for Kevin Bokach, the Senior Communications Officer for the HPTN, and he'll be happy to put you in touch with our speakers today and those also mentioned in the press release. So on behalf of those presenters and the HPTN, I thank you all for being a part of this press briefing. Enjoy the rest of your day. Thank you.